Hello, everybody. Welcome to a new episode of the Dissenter. I'm your host, as always, Ricardo Lopes, and today I'm joined by Dr. Stephen Frank. He is Donald Bren Professor and UCI Distinguished Professor of Ecology and Evolutionary Biology in the School of Biological Sciences at the University of California, Irvine. His areas of expertise include evolutionary genetics, host-parasite interactions, and social evolution. He is the author of books like Foundations of Social Evolution, Immunology and Evolution of Infectious Disease, and the book we're going to focus today, that is the most recent one, Microbial Life History, the Fundamental Forces of Biological Design. So, Dr. Frank, welcome to the show. It's a pleasure to have you on.
Thank you. I'm happy to be here.
So let's start with the subtitle of your book. What aspects of biological design are you most interested in?
Well, really all of it. I think the way in which natural forces have shaped the organisms that we see is the most exciting challenge in all of biology. So whenever you look at an organism and you see that it's doing something, in some sense, I assume that there's been natural processes over evolutionary history that have made organisms that way. And I'm very interested in understanding how those natural processes shape the organisms that we see. It's a real challenge to work out how organisms came to be the way they are. And to me, this is a great challenge in biology.
But what would you say is design from an evolutionary perspective?
Well, just the way in which natural processes have shaped organisms. So it's a very general principle. When you see a bedspread on a bed and it has a pattern and you say it has a design, you assume that in humans, we assume that somebody made that pattern. And the question is, for a biologist, we assume that natural forces have, through time, made organisms the way they are. And so that's the process of design by natural forces. So the question is, how can we understand those natural forces that have shaped organisms? Again, to me, this is the great challenge in biology. And it's not so easy to do. We see an animal has fur and we think, well, the fur is there to keep it warm. And that's fairly easy to figure out. And we generally don't argue too much about that. But fur can have other causes and so on. But some traits are not so easy to work out. And so for a biologist, the challenge is to work out the forces that have shaped many traits that are, again, a bit challenging to understand.
Is design tied to fitness, and how?
Well, yes, I think that's the fundamental principle that essentially all biologists will agree on, even though biologists like to argue a lot. So to an outsider, it may seem very controversial, but it's not really. I think the simple way to think of it is if you look outside the window, I have a window here, I'm looking outside the window. If you look outside the window and you look at the organisms that we see in nature, these are all organisms that have come from a lineage where their parents have been successful at reproducing. Any organism or any trait that's associated with poor reproduction disappears over time, and we don't see it. So everything we see is the outcome of successful reproduction. That's just an obvious fact when you think about nature. Failure to reproduce means that we don't see it.
Yeah. So in the book, you talk about three different fundamental forces of design: marginal values, reproductive values, and generalized kin selection. Could you tell us about each of them?
Yeah, I could say a little bit about those. We're jumping into some really technical aspects of how we work things out. So in some sense, when I just said that fitness or reproduction is obviously important for organisms, because organisms that fail to reproduce and then we don't see them. And so we only see organisms that are successful with reproduction, that is organisms that are successful with regard to fitness. But technically, then the question you just asked me is, how do you actually parse that out when you're studying this as a scientist into particular aspects? And so you just mentioned the three, what I call the three fundamental forces: marginal value, reproductive value, and kin selection. And again, this is a bit technical, but we'll give it a go. Marginal values are a general principle from economics. So anybody that's had any experience with economic principles knows, but actually anybody who has any experience with life, everyday life, knows this very well. Marginal values, let's take a biological example. And since the topic of my recent book is about microbes like bacteria and yeast, we'll talk about bacteria and yeast. And you think about the life of a bacteria, and one of the key things a bacteria has to do is take up sugar. That's the food. So on the surface of a bacterial cell are molecules, we call them receptors, that bind to sugar and bring the sugar into the cell. And so you think one of the things that a bacteria has to do is it has to make these sugar receptors and put them out on the surface and take up that sugar. And so let's think about how many receptors should a bacterial cell make, because it costs something to make a receptor and put it out there to suck up the sugar. So if the cell does not have very many receptors and it doubles the number of receptors, then it can take up sugar twice as fast. And so it has more food and it can grow faster. So that would be a good thing. But eventually, if it keeps on increasing the number of receptors on its surface, it's eventually going to saturate, where if it puts out one more receptor, that's what's called the marginal receptor, it's not going to get very much more sugar. It's already sucking up sugar about as fast as it can. So it's doing everything it can, but one more receptor, maybe a little more sugar, but it's still got to pay the full cost. So after a while, you can see that putting out more and more receptors still costs you a lot, but you're getting less and less back. This is what an economist would call diminishing returns. And so all organisms, when they invest in taking up resources or moving to a new area, or let's say moving faster or slower, will reach diminishing returns. You can put more energy into sucking up sugar, but it's going to get less and less back. So that's marginal valuation. And pretty much everything an organism does is subject to the law of diminishing returns and marginal valuation. So when we think about how organisms allocate their resources to do different things in life, marginal values are crucial. Again, it's just a basic economic principle. And Darwin actually noted that many aspects of many trade-offs in life are really just basic economic principles. So I think that's the first thing that anybody who wants to think about life needs to think about: resources and returns from that economic perspective. So marginal values calls attention to that principle. The second point is reproductive values, a little bit more technical and more specifically biological. But when we think about fitness, the usual argument is you count up the number of babies that you have. And if you have 10 babies, that's a certain amount of fitness. If you have 20 babies, that's twice as much. Or if you were to help a relative, let's say you have some brothers and sisters, and you might help your brothers and sisters, and they carry the same genes that you do. So helping your brother and sister can also transmit your genes to future generations. But then you think about, well, maybe you have a brother or sister who's very young and is just reaching reproductive age. And you have another brother or sister who happens to be quite old and is maybe kind of past his or her prime. And so in some sense, helping one or the other is not the same. If you help your sister who's just coming into reproductive age, then this can be very valuable, because your sister will then, in terms of biological principles of fitness, because she then has a great potential to reproduce, to have high fitness. But if you help a brother or sister who's older and is past reproductive prime, that can help that individual survive, but maybe that individual's not going to reproduce very much. So when we count up individuals and the value of making a baby or helping another individual, we have to take into account what's the future potential reproduction for that individual. And so that's reproductive value. So again, it's kind of like in economic counting. How valuable is this individual, this baby, this relative, and so on? And the value in terms of reproduction is reproductive value is the potential future reproduction of that individual. This generalizes in many ways. You can think about a bacteria that's living in a good habitat and a bacteria that's living in a bad habitat. In the good habitat, the bacteria have high reproductive value because they have great potential for reproduction. And in the bad habitat, there's low reproductive value. So organisms tend to tune their characteristics for the habitats that have high reproductive value. It doesn't do much good to have really good characteristics if the habitat that you're in is never going to be very successful. So this reproductive value weighting is another technical factor that you have to take into account when you're studying organisms. And the final point was kin selection, which I briefly alluded to, which is the idea that you can transmit genes by reproducing, and that transmits your genes and your traits to future generations and shapes what we see. But your relatives, your brothers and sisters, also carry genes that are similar to yours. And so you can transmit genes also through relatives. In terms of microbes, we might say, if you think about two bacteria competing for some sugar, and let's say they're genetically identical because they recently came from a common cellular division. If one bacteria outcompetes its neighboring bacteria by taking up sugar faster so its neighbor doesn't have any sugar, that might allow the bacteria we're talking about to reproduce faster, but it doesn't really gain anything because the bacteria that it outcompeted carries the same genes. They're genetically identical. So when organisms are genetically identical, competition doesn't really improve long-term success in a biological manner. In fact, cooperation tends to be better. If those two can work together to increase their total reproduction, that increases the transmission of genes. On the other hand, if the two individuals are genetically dissimilar, they're not the same, then if one individual takes up the resources faster and outcompetes its neighbors, that causes an evolutionary change because now the focal individual, which was genetically distinct, will have more babies, and the other individual gets outcompeted, which is genetically different, will have fewer babies. And so that changes the patterns of nature that we see. So kin selection is, we tend to think of it as brothers and sisters and aunts and uncles. But for bacteria and for most organisms, it really has to do with genetic similarity. How similar am I to you relative to how similar am I to some other individual? It's a relative similarity. People sometimes make a mistake because they say, well, humans and chimpanzees are 99% genetically similar. So does that mean that what does that mean? But it's a relative similarity. I'm more genetically similar to my brother than I am to you. Yeah. And you're more genetically similar to your brother than you are to me. So a bacteria is more genetically similar to a cell that recently split off from a common ancestor than it is to some other bacteria which may have migrated in from a distant location. So I talked about this for a long time because these are very technical factors, but everything that we see in nature is really shaped by these three fundamental forces. So it's maybe challenging at first to think about these different forces, but the good thing is if you can master these three things, then you really become somewhat of an expert in understanding how nature shapes organisms.
Yeah. In the book, you also talk about a distinction between evolutionary and organismal responses to the environment. So could you tell us about each of those and why it's important to make that distinction?
Well, so you say evolutionary versus organismal responses. That's a technical sounding thing, but it's a very simple point. Basically, if it gets cold out, you put on a coat. That's an organismal response. You're cold, so you do something to protect yourself. You're an organism. That's your organismal response. Mm-hmm. But if you move into a habitat that's going to be a lot warmer, which maybe we are, or you move into a habitat that's going to be a lot colder, and this is going to be true for many biological generations, then the population may evolve to have a thicker layer of fur or to have a thicker layer of fat to protect against the cold, or maybe to learn to sweat. You know, we can sweat, but dogs can't sweat. So dogs pant, but dogs are very bad at handling the heat. Humans are actually much better at handling the heat than are most other mammals because we can sweat and we have various ways of dealing with heat. So that was an evolutionary response for humans to deal with overheating. Maybe not so much from external temperature, but rather early humans were thought to hunt by running long distances and chasing organisms. And human hunters became primitive human hunters, many, in the distant past. One of the reasons they were thought to be successful is that they could run for long distances without overheating because they developed sweat glands, and the animals they were chasing were much faster than they were. And so humans couldn't actually catch them, but eventually the organisms would overheat. And so humans were great hunters because we kept cool. So, right, so if it gets cold, you put a coat on. If it gets warm, you take your coat off. But evolutionarily, you can change the way you deal with temperature by a population, changing its characteristics. It's an evolutionary response. Again, a simple concept, but if you really want to ask questions about adaptation, you have to distinguish what kind of adaptation you're talking about. So that's a very good question.
Mm-hmm. When we observe a particular trait in whatever kind of organism, what is the best approach for us to understand and learn more about the selective pressures that operated there?
Yeah, so that's the essential question, right? And technically in biology, how do you figure out why we see what we see?
Yeah.
And I think the fundamental answer is simple. It's one word: comparison. Comparison is really the key. And so this is well known in evolutionary biology. In fact, there's a part of biology which is called comparative evolutionary biology. Mm-hmm. And we can think about it in the following way. We were talking about organisms that might have a large layer of fat to protect them from cold. But let's think about a situation where we see an organism that seems to have an unusually large, heavy fat layer to protect. We say, well, that's maybe to protect it. But how could we really know the evolutionary forces? Because there's lots of reasons for having a fat layer. Fat provides energy. Fat is protective for temperature. Fat has a variety of hormonal consequences. So you see an organism that has an excessive, what seems like an excessive fat layer. Why? So really the question we are always asking is why? Why do we see that? That's the fundamental evolutionary question. And again, to me, the answer is going to be comparison. So let's work through that. So how can we figure out why that organism has an unusually large fat layer? What we do is we look at a variety of organisms that are closely related. So if we're looking at let's say a cow, well, cows are mostly domesticated, but you're looking at a horse or various mammals that have hooves, and they tend to be four-legged mammals that are what we call ungulates. And so these would be bison, cows, and horses, and things like that. And we see one of them that has a heavier fat layer. What we then do is look at all of its relatives. Here now we mean by relatives, species that are similar, that share a common ancestor. And we look at all of them and we trace back and we see that maybe most of them don't seem to have this excessive fat layer. And so we go back through evolutionary time and we can kind of work out that the ancestor probably did not have this excessive fat layer. So now we see one organism that has the excessive fat layer. And then it happens that among all of these different buffalo and horses and other organisms that are like that, that a couple of others also have this excessive fat layer. So now we have an opportunity to make a comparison, because we can ask, when the organisms moved into a colder environment, was that associated with the evolution of a greater fat layer? And the answer could be yes or no. If we see that every time an organism moved into a colder environment, the fat layer tended to increase, then we have a comparison. Move into a warmer environment, you don't see that. Move into a colder environment, you do. So we can compare moving into colder versus warmer environments. And we have many different independent events because we have many organisms that have evolved independently, let's say, to have this larger fat layer. So we have this comparison. Now, we see that, so we say, well, then it makes sense that maybe this fat layer has to do with colder temperature. But we haven't proved that because maybe colder environments are more demanding for storing energy. And so it's not really the protection from the cold, but rather it's the energy storage that we need. So we would have to do another comparison and look at the availability of food in the different environments, maybe the seasonality. So you need to store energy for seasons where there's more or less energy, food available. So we haven't necessarily answered the question, but we have a way to, we have a new hypothesis. Okay, maybe it's cold and it seems to be consistent with what we see, but maybe it could also be food instead. So we have to do another comparison. So we have a method, which is comparison. It doesn't easily and automatically answer our questions, but it provides us with a scientific way to formulate the question and try to answer it. So that's one key aspect of comparison. But perhaps we'll get to this later, but I'll briefly say that we can also do comparisons on a shorter time scale sometimes. So I see that maybe the next question will deal with this. So let's go on, we'll come back to that.
Yeah. I was going to ask you, so we have different selective pressures. Is it possible for us to predict when we change or there's a change in a particular factor, how it will affect a particular trait?
Yeah, we can predict anything. What we want to do is be good at predicting.
Yeah, right.
And so in the previous example, I essentially predicted that organisms that move into colder environments are more likely to develop a thicker fat layer. And that doesn't sound very profound. Anybody could think of that. And that's a prediction, but it's a reasonable prediction. And a lot of common sense predictions about why organisms are the way they are like that. And so essentially, it doesn't take a lot of a PhD to make a good prediction. It just takes a bit of common sense. But there's a lot of aspects of organisms that are a bit more subtle. And so your question then is really, to me, a question about, can we make predictions that are maybe not so obvious in everyday life about what has shaped organisms? Because a lot of things that organisms do are pretty puzzling, and we don't really easily understand why they do it. And so this goes back to your previous question about what I call the fundamental forces, marginal values, reproductive values, and kin selection. And so to me, the real art of being good at understanding biology is being good at understanding how those fundamental forces, in theory, will shape the traits that we see, and then using the notion of comparison to test those ideas. So that's pretty much the whole story. And just to give an example that makes it, to show you that it's not always quite so obvious, if we think about bacteria and the notion of kin selection. So this is, at first, kin selection in bacteria doesn't seem so obvious. What's a brother and a sister of a bacteria, right? But if you think about bacteria, if they're growing in a region where they're around other bacteria that are genetically very similar, that reduces the tendency for competition to be favorable. Because remember, outcompeting a neighbor that's genetically similar provides really no evolutionary advantage, whereas outcompeting a neighbor that's genetically different. So we can make a comparison. We can compare bacteria that are competing with neighbors that are genetically similar, and we can compare bacteria that are competing with neighbors that are genetically different. So we can run a little experiment. So here's the experiment that we can run, and it's a comparison. So first of all, we start off with some bacteria. These bacteria take up sugar. Once they take up the sugar, they grow. That's what bacteria do. So we have a little container of that, and we put some sugar in there. and we throw some bacteria in there. And with the bacteria that we put in, they all descended from a recent cell by cell division. So they have a common recent ancestor. So they're genetically more or less identical. And they grow up after a while. And then after a while, we have a whole bunch of bacteria because the bacteria have changed the sugar into more bacteria. That's what bacteria do. So we have a whole bunch of bacteria. And so then we ask, well, what's the fitness of the bacteria? It's pretty much the total amount of biomass that's present, because all of these bacteria are more or less the same. Now we could run the same experiment, but we can put in different bacteria that are genetically distinct. Now, if one of the bacteria takes up the sugar faster and grows faster, now competes its neighbor, it will grow up, and then at the end of growth, this is the bacteria we'll see as the one that outcompeted its neighbor by taking up the sugar faster. And the bacteria that was slower tends to not reproduce so much, and so there's not so much of it. So this is a comparison now with a genetically dissimilar organisms competing. And in this case, then what's the success of the bacteria? Well, the bacteria that grew faster, there's a lot of it, so it's successful. But to grow faster, it had to use more energy to make more receptors on its surface to suck up more sugar. So it actually makes less biomass because it's spending more of its energy to basically beat up on its neighbor and to be competitive. Mm-hmm. So the energy spent to be competitive is not available to actually make more cells. So the bacteria can grow faster, but it grows less efficiently. This is advantageous when the bacteria are competing with neighbors that are genetically different. So we have a prediction. If you mix up bacteria with genetically different neighbors, they'll compete with each other by making more receptors and doing other competitive things because they need to grow fast to outcompete their neighbors. So they'll grow fast, but they'll be less efficient. So a given amount of sugar will make less biomass. This is what's called the tragedy of the commons. Yeah. Right? There's a common resource, sugar, but because they're competing, everybody ends up being less successful because they have to expend their energy to fight with each other. And so everybody makes fewer babies, which is the measure of success. On the other hand, if the bacteria are genetically similar, there's no advantage in competing, but rather in cooperating because they're genetically the same. And so they'll grow up, but they'll grow up in an efficient manner. And they'll use the resources efficiently and prudently to grow. And so they'll make more biomass. So basically the notions of efficiency versus competition become fundamental. And we see this is, of course, the great challenge in human life. You know, the problem of solving global warming is in some sense the tragedy of the commons. We all agree that we would like to solve global warming, but everybody else would like somebody else to pay for it. In some sense, it's essentially the same concept here, and it's a very profound and simple concept. But in biology, we can make that prediction and test it. If you test that prediction in bacteria, you'll find that it's true, that bacteria that are competing with genetically different individuals become less efficient and invest more in growth, but actually use resources less efficiently. If you put them so they're isolated with only genetically similar individuals, their traits shift to becoming more efficient and more, we say, prudent. It's a human term, but they use their resources more efficiently so they can transform their resources into reproduction in a more efficient manner. This shapes everything that we see in life, is this tendency for competition versus efficiency. And so these principles that we have, fundamental forces, allow us to make predictions about under what conditions will competition be dominant. And so we'll see organisms that are actually, in some sense, they seem not very well adapted, but what they're adapted to is outcompeting their neighbors. In other circumstances, they're adapted to using external resources efficiently. And we can predict when we can see those different things. And a lot of patterns of nature come down to that. But we have a comparison. That's the key. So we can make comparative predictions.
Mm-hmm. But when we're studying traits, do we approach it differently if we're studying their evolutionary origins or their modification over evolutionary time?
You know, that's really a fundamental question for anybody who wants to understand why organisms look the way they do and trying to understand what evolutionary biologists think. In fact, a lot of evolutionary biologists tend to fight because they get confused about that distinction between origin and modification. I think a good example is when we talk about humans, we often talk about origins. We're very interested in why do humans have such big brains, right? It's an interesting question. We would all like to know why, because humans' brains are relatively larger than other apes that we descended from. Mm-hmm. Why is that? And there's many theories about this. And a lot of the theories, they're good ideas. Again, they're predictions, right? You can make predictions. They're good predictions. But the truth is it's very hard to study evolutionary origins because for humans, we have a sample size of 1. We have humans, they have large brains. The origin of large brains happened a long time ago, maybe once, maybe a couple times, but effectively we just have a sample size of 1. It happened once. We don't have a lot of different human species that we can look at, each of which evolved a large brain, and maybe some of which did not evolve a large brain. So we don't really have any comparison. So studying the evolutionary origins is challenging because you have to find comparisons that you can do about things that happened a long time ago. Sometimes we can do that, but it's hard. On the other hand, modification is much easier to study. If you think about the bacterial example I just gave you, where if you put the bacteria in competition with genetically different individuals, they'll make more receptors on their surface so they can suck up the sugar and outcompete their neighbors. You take those same bacteria and you put them back into a situation where they're around genetically similar individuals, they'll scale back the amount of receptors that they make on their surface because that's very costly and they could be more efficient if they made fewer receptors, they took up the sugar a bit slower, and they grew a bit more slowly and more efficiently. So we can actually see in real time the modification of those traits. To give a human example, there's a gene called the sickle cell anemia gene, which often gets discussed when people are talking about biology. This is a gene that changes the shape of our red blood cells, and it's known that if you have this gene, you can be protected to some extent against malaria. But if you get 2 copies of that gene, you then get a very painful disease that hurts you. And so you get a benefit, but there's a certain cost. And so when malaria is very intense and prevalent, the sickle cell gene tends to increase in frequency because it protects against malaria, even though it has some other negative effects. But other human populations that live where there's relatively little malaria, you see a decrease in the sickle cell anemia gene. So that's an evolutionary modification. It's a comparison. We can see it almost in real time. I mean, we can't quite see the change in sickle cell allele in real time, but we can see populations that have lived in different habitats, human habitats, where malaria is common or not, and see the sickle cell gene frequency changing among those habitats. So we have a comparison about an evolutionary modification that's happening in a short time scale. So for an evolutionary biologist, one of the real great advantages of studying bacteria and yeast is that they evolve very quickly. And so we can see in real time how they adjust. And we can see if our predictions about marginal values and reproductive values and kin selection really work. So that's why my book is called Microbial Life History, The Fundamental Forces of Evolutionary Design, because I'm interested in the forces that shape nature, evolutionary design. But with microbes, we can match the timescale over which the forces act to the timescale over which the changes happen. So we can make those comparisons and in an effective way and really do good science. Mm-hmm. We can talk about why humans have big brains, but it's hard to study. So we may never really figure that out exactly. Again, with a sample size of 1 and something that happened in the distant past, we can talk about it, and we will, because it's fascinating and we love to think about it. But in terms of understanding the forces of nature, it's not a good example because it's so hard to study.
Yeah. But when traits change, what changes first? Is it genes or the phenotype?
Yeah, so this is What changes? Is an organism change, or is there a genetic change? This is, again, a very technical question about modern evolutionary theory, but I'll try to give you some examples. So the puzzle is, when we see, for example, let's think about drug resistance. This is a real challenge, is drug resistance. We all know that if you give too many antibiotics to people, then the bacteria become resistant to the antibiotics, and then the drugs don't work anymore.
Yep.
When you treat people for cancer with chemotherapy, basically the cancer drugs work, but the reason why people don't live forever is that the cancer cells evolve to become resistant to those drugs, right? And so the question is then, so I'm giving you an example of a very general, you asked a very general question, I'm giving you an example,
Yep.
drug resistance. And so how do these organisms or cells evolve to become resistant to these drugs? And there's a long-standing point of view in evolutionary biology that what happens is a mutation arises. So you have some genetic variation, and then you have some force like a drug comes along, and the drug kills most of the individuals, but maybe a mutant is resistant. So you apply the drug, and all the cells that don't have the resistant mutant die, and the cells that have the resistant mutant live. This is genes first. You have the mutation first, you have the drug, and only those cells with the mutation survive. And so this is how organisms change over time. And that's the traditional view. And it certainly is right a lot of the time. We think it's, for example, with HIV, we have drugs that treat HIV, and we know that HIV and people that are unfortunately infected with the virus, they have a lot of virus in their body, and they have a lot of mutant viruses. And when you treat them with the drug kills most of the viruses, but there are a few viruses that are mutated that are already there that resist the drug. And so the drug really doesn't work. And so the way people solve that problem is by treating with combinations of drugs. And so this is what's called combination therapy, which is the success of HIV therapy and why people that are infected with HIV can live. If you use a lot of drugs, simultaneously, there's going to be no viruses that have mutations that work against all of those different drugs simultaneously. And so that prevents then the virus from having a successful mutant, but you have to use a lot of different drugs. So that works for HIV. People try to do that with cancer, and it kind of works, but it doesn't really work. And that's because our cells are different from viruses. Our cells are much more complicated than a virus. And so our cells can respond to drugs differently, or bacteria are also more complicated than viruses. So it's known, for example, for bacteria, if you treat bacteria with an antibiotic, most of the cells will die, but a few of them often live. And so the question is, why do those cells survive the treatment? Is there something special about those cells? And the first thought is, well, they have a genetic mutation, because that's our fundamental principle, our classic evolutionary theory. They have a mutation, so they survive. But you can actually look and see that the cells often don't have a mutation. If you grow that cell up and you make a new population, they don't survive any better than the original population. A few of the cells survive and most of them die. So they actually were not genetically resistant. But what happened was, for example, that some drugs only kill cells when they're dividing. Bacteria tend to divide, but occasionally some bacterial cells stop dividing for a while, just kind of randomly. And eventually they'll start dividing again, just kind of one of those things that happens. So when you treat with the drug, what you're actually doing is the cells that survive are just those that happen not to be dividing at that point in time. But then they start dividing again, they're really no different. So this is a phenotypes first example. So the cells just vary phenotypically, and so they survive. And so it could be that the origin of new traits, drug resistance, starts with cells that aren't really genetically different from the other cells, but they happen to survive. And by surviving, over time, they might eventually get a mutation, and that's phenotypes first. So that's a very technical example. I'll very briefly mention another example which was discussed, which I think is very interesting, which is that there was a goat that was born, that was studied. It turned out that its front legs were damaged.
Mm-hmm.
And so the goat started, from the time it was a little baby, started walking on its back legs. And it became quite good at this, and it could walk around on its back legs, and it got around fine. And it was interesting because the muscles and the bones in the back legs were quite different from a typical goat. And they actually looked a bit more like human legs. They didn't look exactly like human legs, but the muscles and the bones, they differed. This goat was not genetically different from other goats, but it grew up under a different challenge, and it responded organismally rather than evolutionarily. And so this then raises the question, like, one of the big questions in human evolutionary history is, how did humans come to stand up and walk? Because our ancestors, they knuckle walk, right? Chimpanzees and gorillas and orangutans, they really are on all fours. You know, they're using their forearms, their hands and their legs to walk. We're straight up. We're on two legs. How did that happen? And some people think, well, that was a very complicated thing to do, to go from being down, because it changes your spine, it changes your legs, changes your hips. Many things have to change. But if you look at the goat and you think, well, maybe it wasn't so complicated, because maybe if they just stood up and started walking, a lot of their body musculature and bones would have changed without a genetic change. Now, then there would have been some genetic changes to modulate that. So the question is such a change really a complicated genetic change that has to happen, which is hard to imagine, or does the change first happen by the organism changing its response as a phenotype? And so this is a relatively new idea in evolutionary theory. I wouldn't say it's a new idea, but it's gaining more prominence recently, that people understand that it's not always just the genetic change, it's a combination of the two. And so this is a very technical point, but if you really want to understand how organisms change, you have to pay attention to this. And it takes you away from we need to find the gene that changed first and then what happened, which is sometimes a bit more complicated than thinking about how organisms might adjust and then slowly evolve adjustments.
Mm-hmm. And related to the phenotype, what are the conditions that favor the evolution of phenotypic plasticity?
Right, so plasticity just means ability to respond to environmental change. So when I described the goat, its legs were damaged, it stood up on its back legs. We call that a phenotypically plastic response because the organism was flexible or plastic in its development. It had a different challenge, and so it responded plastically or flexibly. And so sometimes organisms range like cold temperature. Some organisms move into cold temperatures and they can grow more fur. Other organisms, they move into cold temperatures and they don't have any way of responding to that immediately. Maybe over evolutionary time, they can evolve ways of dealing with cold, but they don't have any developmentally flexible response. So your question is, why do we see organisms that have this developmental flexibility, which gives them a great ability to deal with different environments? But sometimes we see organisms that don't have that flexibility. And the first answer is, well, having the flexibility is probably always good. So the question might be, well, why don't all organisms have this kind of flexibility? Because it's always good to be able to respond to new challenges. And the answer is probably, we would need to really think about this and test it, but the answer is yes, having the ability to respond flexibly is essentially always good, but being able to respond flexibly takes a lot of machinery. You've got to have sensors which can sense the change in the environment. You've got to have the genes and the physiology that allow a change in response to the environment. And putting together all of those pieces is a costly thing. You know, it's just like saying, well, you know, how many tools do you have at home in your toolkit? The more tools that you have in your toolkit at home, the more things you can deal with if something goes wrong in your home. But you've got some problems because eventually you've got to know how to use all of those different tools, and you've got to buy all of those different tools, and you've got to maintain all of those different tools. And so at some point you go, do I really need all of these different tools? You know, and the answer is no. You know, I mean, you just can't have everything. And so organisms are essentially the same way. They have those tools for things that they are probably going to need to deal with. And for things that they probably won't need to deal with, it's too much cost to get the tool and to maintain it and to learn how to use it and to have all of that extra stuff. And so it's sort of an economic argument. Yeah, it would be good to be able to do anything and to respond to any challenge, but you can't invest all of your resources in preparing for things that rarely happen.
Mm-hmm. When you talk about biological design, is your focus on adaptations?
Yes, adaptations are really the essence of biological design. Just means organisms are designed for their environment. And adaptationist thinking is often considered very controversial, and there's a lot of discussion, both in the popular literature and among scientists, about adaptationist thinking. Is it, you know, and the challenge is interesting because all biologists are adaptationists. All biologists believe in adaptation. Yeah. And all biologists believe that adaptation arises by natural selection. So we basically all think the same thing. And yet, if you were to listen to biologists talk about adaptation, you would think they don't agree about anything because they argue about adaptation endlessly and sometimes in a quite intense and angry manner. And people who are non-biologists often criticize biologists for being overly adaptationist. And so the question really isn't about understanding adaptation. All biologists are adaptationists, and we all think the same way, generally speaking. But it's the fine points. When we see an organism doing something, like a human has a big brain, right? So when you say adaptation, you mean that it's designed to do something. So the question is, why do we have a big brain? What's a brain an adaptation for? And the answer is, I'm not sure. But some people will tell you, oh, well, a big brain is for this. It's for language, it's for feeding, in competitive environments, it's for warfare, it's for cooperation to avoid warfare. You can think about almost everything. Somebody has said it. And so this leads people to think, well, biologists, they're big on adaptation, but then you listen to them and they can't seem to agree on anything. Yeah. But this goes back to our problem. The problem with big brains is that it's an impossible trait to study, but it's interesting to us. And so it's a perfect example of why most examples are bad, which is that if you want to understand how biologists think about adaptation, if you pick the hardest trait that everybody wants to talk about just because we have a social reason for wanting to think about it, then you're going to get a very bad view of understanding biology, which is to say, we don't really know why we have big brains, and it's hard to figure out why. So this leads to a lot of controversy. But when we think about bacteria, why they might compete more for sugar if you mix them up genetically, and we use this notion of kin selection or genetic variation, we have a clear prediction. We understand, we do the comparative test, and we see that it holds, and we understand it. So we have a very clear notion of how the fundamental forces shape adaptations. So the real challenge with adaptation, and the reason why it's controversial, is that there are really three aspects we need to understand. We need to understand the fitness aspect, which are the fundamental force, but you need to understand the constraints, because not anything can evolve. And you need to understand the variation, which was your question about genes or phenotypes. So basically, you need to understand fitness, you need to understand constraint, and you need to understand variation. And when biologists argue, they're arguing about the relative importance of fitness, constraint, and variation for any particular trait. So when you look at a trait of an organism, it can't just grow in any manner. It has to obey the laws of physics. Those are constraints. It can't just, anything just can't evolve. There has to be variation first, either phenotypic or genetic. Phenotypic and genetic variation can only arise if it's possible for that trait to arise. And that depends on the evolutionary history of the organism and on physics. And so when you actually parse out any adaptation, you must always think about fitness, constraint, and variation. Most scientific controversies come down to the fact that people refuse to hold in their mind more than one thing at a time. And so people who really think that constraint is important, they think only about constraint and they argue against fitness. People who are really focused about trying to work out how fitness affects traits, sometimes they forget it. They know that constraints are important, but they tend to emphasize fitness. And so these two groups of people then, they like to fight because people like to fight. Because then, everybody pays attention to them. So it's a human characteristic that humans like to argue and call attention to themselves. But what's interesting is that scientifically, there's actually not really a lot of that we don't understand about thinking about adaptation. What we have are a lot of, the science is fairly clear. The scientists are fairly confused. And so this then leads to people who are outsiders thinking that there's something wrong in biology. But it's the biologists, it's not the biology. That's the problem. The biology is actually fairly simple. Applying it's complicated, but the principles are fairly simple and well understood.
Mm-hmm. And why is it that you focus on metabolism, and particularly in this book?
Okay, so we're jumping from a sort of broad philosophical discussion about adaptation to the book I recently wrote, which is about microbial metabolism. And I chose microbial metabolism for several reasons, but ultimately, my interest is in studying adaptation and biological design. And I've talked about how difficult it can be to study adaptation and design because you need to be able to match your predictions, which are based on fundamental forces, fitness, which is based on marginal values, reproductive values, and kin selection, for example, to make comparative predictions that you can then study empirically by looking at what organisms do. Remember now, the origin of traits like the human brain, they're interesting traits, but they're very hard to study. So if you're really trying to understand how good are we at understanding adaptation, you need to match your tools, which are understanding fitness, constraint, and variation, to the particular problem that you're looking at. Microbes evolve very quickly. So we can match our predictions to comparisons that we can make and actually study. I gave you an example, right? If you mix bacteria with relatives versus non-relatives, genetic mixing versus no genetic mixing, we can predict the degree to which the bacteria invest their resources in taking up sugar faster, which is a fundamental aspect of metabolism, but growing less efficiently because they've allocated their basic fundamental metabolic resources to taking up sugar without competing their neighbor. Whereas if you grow them around genetically similar individuals, they scale back their degree of competition, they take up the sugar more slowly, they grow more slowly, and they go through the biochemical steps of metabolism in a more efficient manner so that they can, for every bit of sugar they take up, they can actually reproduce more. So they're more efficient metabolically. This then connects the notion of kin selection and genetic variation, which seems like a very abstract notion about uncles and aunts and cousins, to actually the way in which microbes adjust their biochemistry to me, this is one of the most amazing things about all of modern biology, because if you're a biologist and you're trained in biology, you get trained in biochemistry, and your biochemistry textbook is this big and it weighs about 50 pounds. And it's all about chemical reactions and about how you take up sugar and you transform sugar into the forces that allow you to run your cells in your body and allow you to run, literally a human to run comes from these sorts of metabolic processes. But there's nothing in this book about kin selection. There's no biochemistry textbook that talks about genetic variation and kin selection and that kind of thing. But what we see is that these reactions of biochemistry can be tuned and modulated by the fundamental forces of biological design. This brings together the sort of most natural history notion of biology. Why do we have big brains? Why do organisms look the way they do? To the most basic biochemical notions of how organisms work. These are things that have been widely separated in the history of biology. And what's exciting to me is that we can talk about them, because we all know that there's nothing in biology that really we can understand without thinking about evolution, but biochemistry does it anyway. Biochemistry effectively hardly ever talks about the tuning of biochemical reactions with regard to the fundamental forces of biological design. So to me, this is one of the greatest challenges in modern biology. And metabolism is important because the metabolism in our body is essentially the same as the metabolism that bacteria have. Biochemically, we're really not that different from bacteria or yeast or other microbes. And so we can then bring together the fundamental forces of biological design, which I say shape everything, and we can study with the most fundamental of all traits, which is metabolism. In addition, microbial metabolism pretty much shapes the geochemistry of the earth, right? If you think about how resources flow through the environment in terms of carbon dioxide and methane and gases and iron and sulfur and all these things, pretty much it's all being done by microbes. I mean, we're only a small factor, and the microbes are doing all of this. And so one could argue that microbial metabolism is the most important of all traits in life, because it's controlling the chemistry of the earth and all the resource cycling. And simultaneously, it's the basis for the biochemistry of microbes is our biochemistry. So it's the most fundamental challenge if you want to understand biological design. So this is why I tried to go after it.
Yeah. So let's talk about some aspects of microbial metabolism that you explore in the book. What are, for example, the trade-offs associated with microbial growth?
Yeah, so the trade-offs are central. And of course, trade-offs are kind of an economic notion, but we've already talked about it quite a bit when we talked about the example of the bacteria growing quickly in order to outcompete their neighbors, or slowly when they're around genetic relatives to be more efficient. And so one of the fundamental trade-offs is growing faster to be more competitive versus growing slower to use resources efficiently. So you can think about if you give the bacteria a fixed amount of sugar, if they grow very quickly and they devote a lot of their resources to taking up the sugar fast and to transporting that sugar into the cell very quickly, that takes energy. And so there's less energy left over for growth. So fast-growing bacteria, if you give them, let's say, a fixed amount of sugar, the fast-growing bacteria, when all the sugar is gone, they're going to have grown less. There'll be less biomass because they'll have devoted some of that energy to growing quickly rather than efficiently. On the other hand, they could grow slowly and efficiently, and so they would transform that sugar into a greater amount of reproduction. So that's one of the fundamental trade-offs is speed versus efficiency. Now that's true but there's trade-offs for almost anything with growth. If organisms, in order to do anything, the bacteria, like they might want to store resources in case there's a period of time where resources are not available. So maybe they want to make what's equivalent of fat. It's not quite fat, but it's an internal storage. And bacteria have internal storage of energy. If they want to store resources, they're going to grow less. So storage is a trade-off that provides the bacteria with the opportunity to survive periods where there's less food available. But in return for that survival, they must grow less in the current time period. So basically, almost anything that the bacteria do other than grow will trade off versus growth because the bacteria have a certain fixed amount of resource. There's many trade-offs that people like to discuss, and so the real challenge is to figure out what the important ones are. A bacterial cell has a limited size, and so there's only a fixed amount of protein that can be in that cell. And the proteins do various things. They do the business of life in terms of making reactions go and building the processes of cells. And so they must, if they're going to do some things with those proteins, like break down food more quickly, then there's less protein left over to do other things, like build up new molecules to make babies. So trade-offs are everywhere.
And another thing that you explore in the book, how do microbes deal with thermodynamics when extracting energy from food? For example, in the book you talk about how the entropy disequilibria in food drives the entropy disequilibria of life, basically. So could you explain that?
Okay, well, you like to go for the fundamental questions, right? So thermodynamics
Of course.
is the fundamental question, and really, I mean, it's a great question. It's a challenging question to answer briefly because you're effectively asking, how does the universe work? Because the thermodynamics is kind of the core of it. So I'll try to give a simple example, and we'll see how well it works. So let's first back up a little bit and ask basically how do forces drive change in general? And so if we can let's think about an example. We have a weight, okay? And we have a rope attached to the weight, and we're holding this weight off the ground. So we know that if we let go of the weight, it's going to fall to the ground, right?
Yeah.
So that's just a basic principle. Everybody agrees with that. And if we actually connect this rope to some other thing, like a wheel that turns, then as we drop the weight, it can turn the wheel and do some work. And so what's really going on there? Well, the way to think about that is that the weight off the ground is in disequilibrium. Disequilibrium just means that it's going to naturally move if you just leave it alone. It's going to naturally move to a state where it doesn't change. But if it's held off the ground, it's in a state where it will change. Right? So that's a disequilibrium. Being on the ground is an equilibrium. If you have a weight sitting on the ground, you can't do any work with it. So it isn't the weight that does the work. It's the disequilibrium of the weight being off the ground that can allow us to use that weight to do work. And so basically everything in the universe that is a process where there's work being done or something being driven or changed is you're taking one disequilibrium, like a weight off the ground, and as it moves towards its disequilibrium, you're capturing that to drive something else to make that into disequilibrium. So you're transforming one disequilibrium to another, which is roughly the definition of an engine. And so everything in the universe where you're seeing order being created or work being done is basically taking one disequilibrium and capturing it to create another disequilibrium. So it's the transformation of one disequilibrium to another. So that's the most generic principle. And if your listeners listen to this, what I just said a couple of times and think I kind of understand that, then that's fantastic, because that's the most fundamental principle of all of science, is that nothing happens without one disequilibrium being captured to transform another disequilibrium. So now if we think about what cells are and what food is and how biology works, it's the same principle. Food is basically electrons, molecules with electrons that are in disequilibrium. Now they're not held off the ground, but they're roughly speaking, it's kind of the same thing. They're in a state that's sort of hot, you might say. Now food doesn't feel hot to you, but it's the notion that they're in some state that's not stable. And so food is basically electrons in a relatively unstable state. And what your body does is it takes up that food, like sugar, and it takes those electrons, which are not really in a very stable situation, and it allows those electrons to flow to a more stable situation. So basically the electrons are, roughly speaking, they're off the ground, and your body lets those electrons flow to a more stable situation, but it does it in a way that it captures the change, right? If you just take a weight, you drop it to the ground, well, that's the end of it. You haven't captured anything. You need the rope tied to something else to capture the work. So your body is basically tying a rope to those electrons, and as those electrons flow to a more stable situation, it's allowing the cells biochemically to capture that transformation. So some people may have heard of the molecule ATP, and you know that the molecule ATP is the biochemical molecule of energy. But really, what's really happening is that when your cell takes up some sugar, which is the most basic kind of food with electrons that are in disequilibrium, then metabolism is the process of transforming that sugar through a series of biochemical reactions where the electrons are slowly coming downhill to a more equilibrium state. And with each transformation, your cells occasionally capture some of that change and move towards an equilibrium by taking another molecule called ADP and changing it into ATP. Mm-hmm. So what your body is doing is transforming ADP to ATP. So that's a molecule with 2 phosphates to a molecule with 3 phosphates, diphosphate to triphosphate, adenosine diphosphate, adenosine triphosphate. So ATP itself doesn't really contain energy. This is the biggest mistake in all of biology. It's not that it contains energy. What's happening is that there's 2 molecules, ATP and ADP. And what your body is doing is transforming ADP to ATP and making a disequilibrium, a chemical disequilibrium. That's what a battery is. A battery, an electric battery. An electric battery is a chemical disequilibrium. And as the battery moves towards equilibrium, it's possible to capture some of that change and move towards disequilibrium, of disequilibrium moving towards equilibrium, to capture it as electricity to drive work. Your body is making a chemical disequilibrium between ATP and ADP and using it as a storage battery. And then later, when it transforms ATP back to ADP, which is a move to equilibrium, it's using that move to equilibrium to drive another disequilibrium. Remember, all work is a drive towards disequilibrium. When you want to make the molecules that make up your body, mostly what you have to do is drive them away from their natural equilibrium state. Because you all know that when you die, your body decays into an equilibrium state and your molecules decay. So your body is actually held in disequilibrium. So what your body does is takes the disequilibrium in food, which is electrons held in disequilibrium, and it drives the disequilibrium of ATP versus ADP. And then it uses the disequilibrium of ATP versus ADP as a storage battery. And as it dissipates that disequilibrium, it drives other disequilibria to make the molecules of your body or to allow you to run or to do other things with your muscles. And so, although that was a very long answer, that's basically 99% of what you need to know about biochemistry and thermodynamics and how the universe works. So it's very important. And so this was a fundamental point of my book, was to bring these notions of biochemical thermodynamics and connect them up with metabolism, and then connect them up with the principles of biological design, which remember are very abstract principles like economics and marginal values and kin selection and genetic variation. But what we see is that we've brought together all of these different principles, and we can really do this. This is modern science, is some of the breakthroughs in the last few years in modern science at a technical level, which we're not talking about today, allow us really to do this from the biochemical level all the way up to this abstract theoretical level. And it's really the first time in biological history that we can do that, in the study of biology, that we can do this. So it's a very unique time and a great opportunity to bring these concepts together from very different fields, because the technical advances of modern science allow us to make measurements about these things and test ideas.
Mm-hmm. And what is flux and how do cells modulate it?
So flux is just a technical term which means flow. And so when I talk in the book about biochemical flux, really what I'm just talking about is flow. And remember, everything's about disequilibria and equilibria. And so what we're talking about is, when we talk about the flow of electrons through metabolism, the flux of electrons through metabolism, then we're really talking about the flow of electrons from a state which is in disequilibrium to a state which is closer to equilibrium. And then that electron flux happens through various biochemical transformations, which transform one molecule into another, which is a flux. Mm-hmm. Flux is the transformation. And so there's just this natural way of doing things. You can think about the flow from disequilibrium to equilibrium. I said that's not really, it's really that really drives things, rather than this notion of energy. And this is a big mistake people make, and I use the word energy also, but it's really not correct in the way that it's used in typically in biology. Really what's happening is that the electrons are in disequilibrium because they're concentrated energy. And concentrated energy is a bit like heat. When something is hot, it's concentrated energy. And when it cools off, the energy spreads out. And so basically, chemical reactions that go from disequilibrium to equilibrium are concentrated energy transformed to energy that's spread out. So it's like heat cooling. So actually, energy doesn't get used up. Remember, actually, one of the fundamental laws of thermodynamics is that energy is never created or destroyed. Mm-hmm. So when we say, how much energy did you use today when you ran? Well, the answer is you didn't use any energy. You took some concentrated energy and you dissipated it and you spread the energy out. And so it's a flux in terms of energy is concentrated energy moving towards dissipated, distributed energy, a flux of electrons in what's effectively a concentrated energetic state, and allowing them to flow to a less concentrated state from disequilibrium to equilibrium, because heat naturally tends to spread out and cool. So flux is just a term that means flow, and it's the flow from disequilibrium to equilibrium at all different levels that's really crucial.
Mm-hmm. In metabolism, what is the role played by enzyme catalysts, and on the one hand, and on the other hand, changing biochemical conditions?
Right, so that's crucial for actually understanding biochemistry, because our cells, we know, have lots of catalysts that do things biochemically. And so what we need to understand a bit about, one of the key aspects of biochemistry is, let me take an example which is not biochemical first, which is, think of a diamond. A diamond is just carbon atoms.
Yeah.
And they're arranged in a particular way.
Mm-hmm.
And graphite is carbon atoms, right? Graphite's sort of like, the black coal-like stuff.
Yeah.
Not exactly, but that's sort of the material, or like in a pencil, if you're mm-hmm. old enough to remember pencils and you don't only look at your computer. Right, that's just carbon. So diamond is carbon and graphite is carbon. And actually, we might think that diamond is somehow more stable than graphite, but that's actually not true. That diamond is less stable, is in disequilibrium relative to graphite. But that doesn't sound right because you never see diamond changing into graphite. And on the conditions that occur in the surface of the Earth, diamond would eventually change into graphite, but it would happen so slowly that effectively it would never happen. I don't know how many an incredibly long period of time it would take. I don't know the right answer. Is it billions of years or something? It happens so slowly that it doesn't happen. So diamond is in disequilibrium relative to graphite, but that chemical reaction is not going to happen. So our bodies are molecules that are in disequilibrium because when you die, your body's going to decay. And decay basically means your molecules are going to move towards their equilibrium, which is going to be simple, molecular forms, whereas you have a variety of complex molecular forms. So your body's in disequilibrium. And so what your body does is basically uses chemical catalysts to control rates of reactions. It uses disequilibria to drive some molecules into a disequilibrium state. And then sometimes it needs to break down those molecules, like proteins are in disequilibrium, because proteins will eventually break down into simpler molecules. And your body can break down proteins by chewing them up, and it uses catalysts to do that. So what a catalyst does is it takes a reaction that would have happened, but maybe very slowly, and it makes it happen more quickly. So although I don't know of any biochemical catalyst that can make diamond into graphite, it's the same idea. You can take a protein, like you eat a protein, you eat some food, it has protein. That protein might take a very long time to break down into simpler molecules, but your body has catalysts that take those proteins and they break them down into simpler molecules. Then it has catalysts along with disequilibria like ATP versus ADP, which build up other molecules. And so catalysts are basically changing the rates of reactions. And so your body is constantly modulating the rates of different chemical reactions to break some things down, to build other things up. Sometimes that has to do with, saving up disequilibria for later use. And sometimes it uses disequilibria to create molecules that are more ordered. So that's basically all of how biochemistry works.
Yeah. So just one set of two final questions before we wrap up the interview. The principles of biological design we've talked about before. Are these the only principles out there? Is there a fixed set of principles, and do they apply across the biosphere or only to microbes?
Well, to answer your last question, the principles of biological design apply to all organisms. And remember, in general terms, there's no controversy. All biologists essentially all biologists agree that organisms have been shaped by natural selection, and that everything we see in terms of organisms being able to adapt to the environment and achieve most of their functions arises or is shaped by natural selection. Natural selection is an extremely easy idea. It's basically trial and error learning. You try some things out, the things that work well you keep, the things that don't work well you get rid of. So that's the fundamental biological principle, and it applies to all of life. But actually using the principle of natural selection to study biology, so that applies to all of life. Breaking that principle of natural selection into things like marginal values and reproductive values and kin selection is something that I would say are general principles that apply to all of life. Other biologists like to argue about that, but in fact, I would say there's relatively little argument about it, really. Those really are the principles, and the arguments really have more to do with how do we formulate and test specific predictions, and when are we convinced that an explanation is correct, and when do we pay more attention to constraints or the origin of variation? Because remember, there's always fitness, constraint, and variation. Fitness is the principle of differential reproductive success. Constraint is that organisms must obey the laws of physics. And so organisms can only do what is physically possible. And physics really constrains a lot of what organisms end up looking like and doing in life. And variation is that organisms cannot run trial and error natural selection if there isn't a trial to be run, which is a variant that does something. So maybe humans would be better if they could have a particular trait, but unless there's a variant of humans that has that trait that could possibly spread in a human population, then humans will never acquire that trait. The variant has to arise. It has to be physically possible. It has to be biologically realized. So there is a simple set of biological principles. In my view, the principles that I'm talking about, they're my personal view, are the simple principles that explain most things, and we can apply them to microbes, but we can apply them to all organisms. And our fundamental tool is comparison. That's the way we study whether our predictions are good predictions. And the way we make good predictions is using the key aspects of natural selection, constraint, and variation, and thinking carefully about those different factors.
Mm-hmm. So the book is again Microbial Life History, the Fundamental Forces of Biological Design. Just before we go, Dr. Frank, would you like to mention where people can find you and your work on the internet?
Well, my website, which is at www.stevefrank.org. Just type in Steve Frank Evolution into Google and you'll find my website. And on my website, you can actually download my prior books, several prior books for free. The new book is not available yet. It hasn't been published. It'll be out in August, published by Princeton University Press. There's the introductory chapter and table of contents for the new book is on my website. So you can download that and see what's available and where the work is going. But in summary, thank you very much. Your questions were very deep and penetrating. You asked some of the most challenging scientific questions. And so I hope that your listeners would at least get something out of the various parts of our discussion. So thank you.
Yeah, thank you so much for the kind words. Thank you for your time, and it was really fun to talk to you.
Thank you. Appreciate it.
Okay, and it was really fun to talk to you.
Thank you. Appreciate it.
Hi guys, thank you for watching this interview until the end. If you like what I'm doing, please share it, leave a like, hit the subscription button, all of those things you already know. And please consider supporting the show either on PayPal or Patreon. All of the links will be in the description box of the interview, starting at $1 per month, so it would be a great help. This show is brought to you by Enlights, Learning and Development Done Differently. Check their website at enlights.com. I would also like to give a huge thank you to my main patrons and PayPal supporters: Karen Litske and Blanchett, Pererga Larson, Lau Guerrero, Francis Ford, Hans Frederick Sunder, Ricardo Vladimiro, Craig Healy, Adam Castle, Olaf Alex, Jonathan Wiesel, Jacob Klinkby, Matthew Whittingbird, Arno Wolf, Tim Hollisey, Eric Alenia, John Connors, Paulina Barron, Philip Force Connolly, Jerry Mueller, Herbert Gintis, Rutger Vosbo, Weingard, Rebecca Newberger Goldstein, Dan Demetrio, Robert Windager, Rui Narcio, Arthur Coe, Zup, Marco Neves, Colin Holbrook, Susan Pinker, Bernardo Seixas, Pablo Santurbano, Simon Colombo, Jorge Espinha, Phil Kavanagh, Mark Blythe, Roberto Inguanzo, Michael Stormir, Eric Neumann, Samuel Andreev, Tiago Nunes, Bernard Ugni, Alexander Dunbauer, Fergal Cusson, Evan Bodrenko, Al Herzog, Don Ross, Jonathan Librant, Oslam Bulut, Nathan Nguyen, Stanton T, Samuel Correa, Eric Hines, Mark Smith, J.W., João Eira, Tom Hummel, David Sloan Wilson, Yacila Dez Araújo, Eden Solon, Romain Roach, Dmitry Grigoriev, Diego Lononio Correa, Tom Roth, Yannick Punter, Adana Rusmani, Charlotte Bliss, Nicole Barbaro, Adam Hunt, Pavel Ostazewski, Nelek Bach, Catherine and Patrick Tobin, Al Ortiz, Guy Madison, Gary G. Hellman, João Linhares, Lida Cosmidis, Saima Afzal, Adrian Yagi, Nick Golden, Paulo Tolentino, João Barbosa, Jules Price, Edward Hall, Edin Bronner, Franca Bortolotti, Gabriel Pons Cortez, Ursula Litske, Denise Cook, Scott Zachary Fish, Tim Duffy and Trader in NYC. My producers Isar Webb, Jim Frank, Lucas Stafiniak, Ian Gilligan, Luís Caetano, Tom Vanagdam, Curtis Dixon, João Linhares, Benedict Mueller, Vega Guidi, Sardus France, Thomas Trumbull, and Nuno Welder, and my executive producers Michel Rugieski, Rosie, James Pratt, Matthew Lavender, Sergio Quadriano, and Bogdan Kanivets. Thank you for all.